Thank you, Russ, for that very wonderful and warm introduction. My name is Dr. Tho Pham. I'm one of the Chief Medical Officers at the Stanford Blood Center, and today I'm very happy to talk to you about methods and practices that we currently use to reduce and prevent transfusion-transmitted infections. And what I really want to do is kind of pull back the curtain on some of the things that we kind of do behind the scenes here to make sure that the blood that we collect from you and produce from that is as safe as possible. So here's an outline of the key topics we're going to land on tonight. First, we'll talk about just the current state of affairs of transfusion-transmitted infections and put it into a historical perspective. Then we'll go into the current testing methods that we're currently using, and you can break it into two major pots after that. It's nucleic acid testing and also by serology testing. And then like everything else, there's always going to be areas for improvement. So in addition to those tests, we'll talk about, despite those tests, we'll talk about places where there might be some blind spots, so things such as bacterial contamination and also potentially emerging infectious diseases. And lastly, we'll just talk about a couple of things that are that we're currently doing to proactively curtail those blind spots that the testing methods don't really address all that well. One is pathogen reduction technology that we're very proud to speak to, and the second is a very nascent research project that I've been involved in using a linear accelerator to sterilize blood. So let's go. So first, current state of affairs, just where we are right now and where we've been. Right. So just some basic numbers, right? Just to give you the scope of the scale of transfusions in the United States. First, there's about, annually, about 17 to 20 million units of blood products transfused in the United States every year. And so about 12 million or so are red cells, and another 3 million are platelets, right? So of those, these are all provided by about 13 million donors annually. So people like you who come in and donate all these blood products to all these patients who would otherwise either die or be severely hurt or maimed without it. So once again, take this quick moment to thank you again for all the donations that you have provided to all the patients over the years. And all these blood products currently on the shelf and all these transfusions currently being done, in the millions. It's very important to make sure that the blood is as safe as possible, right? And we have to go, 1% is too high. 1 in 1,000 is too high. If you're transfusing 17 million units a year, 1 in 1,000 is still 17,000 events. So you want your blood very, very safe. And over the years and decades, we've gotten that safety level as high as possible. The CDC says that the U.S. blood supply is safer now more than ever before, which is good. However, nothing is 100%, nothing is 0%. So despite the high level of safety that we have now, there's still a small but finite risk for transfusion-transmitted infections, and these come from bacteria, viruses, or any other parasites and pathogens. And we'll go over that. So the current risk right now of TTIs, or transfusion-transmitted infections, can really be summarized with this table, right? There's this column here represents the different viruses and pathogens. So you have HIV, the hepatitis viruses, HCV, HBV, HTLV, CMV, West Nile virus, Zika virus, Treponema pallidum, various bacterial species, Plasmodium species, Babesia, T. cruzi, and variant Creutzfeldt-Jakob disease. Okay, and this next column tells you the clinical symptoms how they manifest. Obviously, AIDS from HIV, hepatitis from the hepatitis viruses, and then T-cell leukemia from HTLV. CMV can give you either systemic symptoms or more localized symptoms to the GI tract or the retina. West Nile virus can give you a very severe neuroinvasive disease in a small percentage of patients who get infected with it. Zika virus, as is notoriously known, is associated with microcephaly as part of this congenital Zika syndrome. And also in other adults, there's been a correlation of Guillain-Barré with Zika virus. T. pallidum is the causative agent of syphilis. Bacteria, various type of bacteria can give you, various types of clinical manifestations, but the one noted here is sepsis. And of course, going down the list is malaria, babesia, Chagas disease, and of course, transmissible spongiform encephalopathy. This column tells you the current risk. Now, you can see that for almost all these, the risk is basically 1 in some millions. So the risk is very, very low. And all these honestly are calculated numbers, right? So they're all very low risk, 1 in some millions. And there are some in here that basically there are no risks because we haven't seen it. It's only theoretical, but we still test for it or still mitigate it somehow. The one exception is going to be the bacteria sepsis from platelets, and that risk remains at 1 in 100,000. And we'll get to that in a little bit. So that's where we are right now. Really, really impressive numbers, right, as far as like safety risk. But where did we come from? So this table kind of shows that. The x-axis is the year, time in years, and then the y-axis is the risk of a unit of blood being contaminated with one of these 3 viruses, HBV, HCV, or HIV. As you can see, back in the early 80s, the risk of having a unit contaminated with HCV virus is, looks like close to 1 in 50. That's a very high number, right? And then you can see that also with HIV, it peaked in the 80s at close to 1 to 100 level risk. And similarly, HBV was high there too. So back in the 80s, the risk of a contaminated unit was quite high. And since then, that rate of contamination has dropped, and you can see that steady decline to where it is now, where it's close to basically in the realm of 1 in a million, right? And a lot of it has to do with like sort of awareness that over the decades that blood can be a vehicle for many of these infections, and also there's a lot of donor screening questions that I'm sure you're all familiar with that help prevent this, that help sort of prevent a lot of these transfusion-transmitted infections. Okay. Also, of course, in addition to awareness and donor screening, there's a lot of testing that's been done. And this bottom graph shows the timeline is on the x-axis, and then you can see it. It's like the various arrows represent the various tests that were initiated at that time to screen blood. So you can see that through the years, more and more tests have been piled on, and concomitantly, you see that the risk of infection, as shown in the top graph, goes down over time with the test that's increased. So testing is a very big deal, right? It's a very large part of how we mitigate the risk of TTIs. Now, the other thing with the bottom graph is that you can see that we can't. There's an upper limit, right? Like every time you put on a test, there's resources poured into making that test and doing the test. So with more pathogens on the horizon, it becomes sort of unscalable if you test for every single one, every single bug, every time a new virus or pathogen arises, right? And we'll get to that in a second. So let's go over the current testing methods, right? The current testing methods, I'll break it up first. What are the bugs we test for? So these are the bugs: HIV, HBV, HCV, West Nile virus, Zika virus. And note about Zika virus is that we will cease testing for Zika virus later this year, and that's primarily because as more epidemiological data has been accumulated over the last few years around Zika, has become more apparent that Zika is not a real threat to the blood supply. It has not, sort of been as widespread, luckily, in the United States as we had once feared. Babesia is something that some blood centers on the East Coast, where that disease is a little more prominent, test for. T. cruzi, Treponema pallidum, and HTLV1 and 2. So those are all the bugs that we currently test for by one method or another. Now, as far as the methods, there's really 2 main methods by which we test for these bugs in total, right? On the left is NAT, that stands for nucleic acid test. And we'll get to that in a second. So the nucleic acid test, you see, we test for HIV, HCV, HBV, West Nile, Zika, and Babesia. And what that's really doing is looking for signature sequences of nucleic acid of that pathogen in the blood. So really what you're testing for is, can the bug be detected in your blood at this moment right now in time, right? And so that's what nucleic acid is. And now serology testing, on the other hand, is sort of like looking for antibodies, or in this case, looking for proteins, and in this case, specifically antibodies. And the antibodies, if you have antibodies against a particular pathogen, it just means that you've been, sometime in the past, you've been exposed to it or infected by it. So it really is like the question, so what I was really trying to answer is, have you been previously infected by the bug? Not necessarily are you currently infected, but have you ever been infected by the bug? So there's some sort of nuanced differences between those the questions that these two tests answer. Okay, so let's start with we'll go through some of the general principles underlying these tests. So first, let's do the nucleic acid test, right? So nucleic acid test. So let's talk about that. The first is, so you see on the left, you see a stereotypical prokaryotic cell. So it's a bacteria or something like that. Within the bacteria, you have its genetic information, its DNA. So the thing about DNA or RNA is that it provides the genetic code, the blueprint of how to make the organism, right? But another way to think about it, this is how all organisms propagate, right? You have DNA or RNA, the blueprint to make you, and the way that you propagate or replicate is to make more of that, so that it can make more of you, right? Very simplistically. And the good thing about the thing about DNA is that it's like a very long strand of a series of nucleic acids. And in this case is represented by 1 to 4 letters, C, G, A, or T. Okay, and the string of nucleic acids in an organism can be extremely long, but the fascinating thing is that because the string is so long, and you can probably find a specific spot in their chromosome, in that genetic code, where the string is unique enough to that organism that that a string of certain letters side by side represents this pathogen. It's a natural barcode for you to be able to identify a specific pathogen if you know its genetic code, right? So that's very powerful, and that general principle underlies the testing that we do. And the main, in a nutshell, one of the main sort of ways that we test for it is by a method called PCR. It stands for polymerase chain reaction. PCR, you hear it all the time, right? So first, let's just say this is your blood, and then you see here that there's this pathogen, the DNA of the pathogen floating around your blood, and is represented by this double-stranded DNA here. Now, if you know the sequence of the bug, what you can do is you can design primers that target, that are specific to that pathogen, that are barcoded for that pathogen, and it targets that pathogen's DNA, right? And then, so now it's specific for that. And the next step, what you do after that is, through a series of opening up the DNA, letting the primers hybridize, basically find its spot on the DNA molecule, and then letting it replicate. What you essentially do is, after one round, you have one molecule of DNA turned into 2 molecules, and then you repeat the process again and again and again. After 2 cycles, you have 4 molecules of DNA that originally start with 1. After 3 cycles, you got 8, et cetera, et cetera. So the nth cycle, you have 2 to the n copies, if you start with 1. So we normally do 30 to 40 cycles, right? So to put in that perspective, if you start with 1 molecule, after 30 cycles, you have 1 billion molecules of the sequence. And so it's really, all this is really just to amplify up the original genetic material that is found in your blood of that pathogen. So once you amplify it up, you have to detect it. You amplify it up to a high enough level that you can detect it. And the way you do that is like usually for many of these technologies, for every DNA molecule that's sort of amplified, there's a sort of fluorescent probe that is attached to it. So it lights up. So the more DNA molecules that you make at the end of your 30 cycles or whatever, the brighter it gets, and that can be detected by a machine, in essence. So that's really the underlying technology of pathogen detection by nucleic acid technology. You have nucleic acid specific sequences of the pathogen in your blood. You amplify it, you label it, and then you detect it. And if you can detect it, then you know that that bug is there. Now, moving on to the other side of the house for testing is serology testing. So in general, serology really, it's our immune system, the human immune system, or the immune system of many jawed vertebrae. You can develop antibodies against almost any foreign pathogen, whether it be almost anything foreign. It doesn't even have to be a living thing, honestly, in many cases. So, and the thing is, when it develops these antibodies to the pathogen can be very specific because it goes through more or less like a microcosm of an evolutionary process within your body, so that the antibody gets more and more sort of specific for that pathogen. So eventually, you have a basically lock and key pattern where this antibody is specific only to this pathogen, right? There's always a small possibility of cross-reactivity, but nothing's 100% in biology, but the specificity is extremely high. The small possibility of cross-reactivity is really one of the things that underlies false positive results that many donors get. The other thing about the serology, as opposed to the nucleic acid, is that this process of making antibodies against a foreign pathogen, in general, it takes about 2 to 3 weeks to develop, right? So the fastest time that you can sort of detect an antibody against a specific antigen in a person is biologically defined as at least 2 or 3 weeks after they've been infected. So the first 2 weeks or so, even though they're infected, by serology methods, you won't be able to detect anything, okay? So the biology that underlies serology tests is really about how B cells detect viruses and then make antibodies against them. So first you start with a naive B cell, that hitherto has not seen a pathogen, a foreign pathogen. And in this picture you have all these sort of squiggly things that are supposed to represent various pathogens that your B cell, your body can see. Once it sees it, the next thing that does a few things. First, it can be activated by it, and then it can start it can differentiate into like a small-lived plasma cell, which can start immediately pumping out, pathogen-specific antibodies in the form of like a couple of isotypes here, IgM and IgG, okay? And like I said, this will take about 2 to 3 weeks to develop, right? Now, a separate but parallel path that happens to many of these B cells that are exposed to the virus is that they go into your lymph nodes, go into the germinal centers in your lymph nodes, they interact with other cells in your immune system, such as T cells, and through that process, many of those B cells can then differentiate into sort of these longer-lived memory B cells that, as the name suggests, keep the immunological memory in your body that you've seen this virus, so that what happens, you have a long-lasting level of antibodies against this specific pathogen, or in addition to that, you may have a subset of B cells that are long-lived memory inside your body somewhere, so that the next time you see it, it doesn't take you 2 or 3 weeks to develop an antibody response. Okay? So it's because of this long-lived memory response that a lot of times, your exposure to a particular pathogen, you have these antibodies in your blood and can be detected for years and sometimes decades after that initial event of being infectious. And so that's why that's the principle that underlies the fact that serology testing tests for whether or not you've ever been infected, or at least like sometime in the past, some long time ago. So between those two tests, we and a variety of other so between those two testing methodologies for those bugs and a variety of other sort of mitigation factors that we use in donor screening, we've been able to get the blood safety to a very high level as it is right now. But like I mentioned, there are still certain areas that we can always improve on, right? So one of the first things is let's go back to this table that I showed earlier. There's the risk of the current TTIs, right? One of the things that stuck out, I mentioned previously, is the bacteria. Bacterial contamination and then resulting sepsis from that is estimated to be about 1 in 1,000, 1 in 100,000 units of platelets. So why platelets? Well, platelets are unique among the blood products because it has to be stored in a very peculiar way. It has to be stored firstly at room temperature, and on top of that, you have to keep it, shaking gently during its storage time. So these conditions, honestly, are perfect to grow bacteria that was originally in the bag, maybe in a small amount before that. I mean, just think about how your food goes bad when you leave it out overnight. I mean, that's essentially a similar thing. Okay. So, bacterial contamination and how that could result in significant clinical events. So what's the rate? What's the current going rate of bacterially contaminated units? Well, that number is kind of out there, and there's many studies that kind of look at this. But so this is one of them that calculates like the number of contaminated bacterial units per 1,000 from apheresis platelet collections is about 0.23. Translated to another way, converted to another way, it's really 1 in 5,000. So that's kind of like a very high number, right? Like 1 in 5,000 units has some level of bacterial contamination. Here, we, at least, about 15,000 units of platelets are transfused a year here. So 1 in 5,000 means 3 units every year have some bacterial contamination. Now that doesn't necessarily translate. It definitely does not always translate into a clinically significant event such as sepsis or otherwise. So the question is then, well, then how, if 1 in 5,000 is contaminated, what's the rate of clinically significant events? So there are various other studies to get to that number. That number is very hard to get to because there's a lot of underreporting potentially and not a lot of uniform reporting methodologies. But there are some studies out there that try to sort of estimate that, and from this, it's estimated to be about 1 in 25,000 transfusions, which is still a high number. But some experts feel that this is an underestimation for the reasons that I just mentioned before. So you got contamination, clinically significant events, and then the next morbid step after that is really fatalities, right? So how many people die from this per year? You see here, like this is in 2018, and it's reported to the FDA. 6 people in 2018 died due to sepsis from a bacterially contaminated unit of blood. And in general, it's about 1 to 6 deaths per year from bacterial contamination from transfusion, about 4 deaths a year. And then, but if you break it down by product, you see that there's an overrepresentation of these contaminated units leading to sepsis and death in platelets, and that's because of the way that we have to store it, because that's the appropriate way to store platelets to make it functional and viable for transfusion purposes. So that's one potential area for improvement, right? Is with these bacterial cultures. Or with bacterial contamination. Despite all the testing that we do and the culturing that we do to sort of minimize the risk, this is still sort of the standing risk that's related to platelet units. The other thing that can use area improvements is, obviously we test for certain specific bugs, but what about new bugs that come out, right? The most recent example obviously is COVID, but what about Ebola and all these other things? New things are always coming out, and the world is getting smaller and more social, so these things can spread a little more quickly. So, and like I mentioned before, you can't possibly test for everything under the sun because it's just not feasible logistically and from a resource perspective. Now, in regards to emerging infectious diseases, there are, it's been broken down. It's been proposed to break down to basically like a tier system, right? So there's a red tier system where, there are these pathogens out there, and we know they are dangerous, and we know that they have capability of being transmitted through blood. And then there's an orange one where It's dangerous, but there's some evidence that it can affect blood safety, but it's not as high priority as red. And of course, then there's always a yellow that there isn't good evidence yet, but it's a possibility, and we'll keep an eye on it. So that's just kind of like a rubric that was developed by a working group. And just to give you an example, like some of these on the red is dengue is in the red category. Babesia, there's a test for that now, but Babesia is considered in the red. And then orange, there's more, right? There's chikungunya, St. Louis encephalitis virus, leishmania, and plasmodium. That's malaria. We don't test for it right now, right? So those are sort of the areas that can use improvement, all right? Bacterial contamination and emerging infectious diseases. So a couple of things we're going to bring up now is hopefully sort of address one or both of those issues. The first is pathogen reduction technology. Now this is something that Stanford has been at the forefront of implementing, and we'll go through that right now. So what is pathogen reduction technology? So PRT for short, and it's considered to be a proactive method for reducing the contamination rate in platelets, right? And it's also effective, been shown at least like in laboratories to be effective against most clinically significant pathogens. So how does it work? So it goes back to nucleic acid again, right? And preventing that nucleic acid from propagating, okay? So first you start with a small molecule. Here it's called amotosalen. And that's sort of like their secret sauce, or I guess it's not so secret because we know what it is, but that's sort of the secret, the main ingredient. And what the amotosalen does, like its characteristics is actually to basically find, or it has a high affinity for nucleic acid, either DNA or RNA, and it will find it if you mix it into a good enough concentration in a particular solution, right? Once it sort of finds it or intercalates, insinuates itself in the nucleic acid, then what you can also do is, the next thing that you do is you illuminate it with some UV light. Here, UVA. And the UV light will activate the amotosalen so that it will become very reactive to everything around it. And what that essentially does, like it cross-links itself with the neighboring nucleic acids that it's sort of attracted to. And that way it's a covalent bond and it can't break free. Now that you've done that, the nucleic acid that's there of the pathogen has no way of replicating. And if the nucleic acid material of the pathogen, i.e. its blueprint of life, can't replicate, the pathogen itself cannot replicate. It can't make any more kids because the nucleic acid underlying that, it cannot replicate. So that in a nutshell is sort of how the PRT system works. Now operationally, it's it takes some sort of training and getting used to, but we did it here. And we implemented PRT pathogen reduction technology on our platelets back in May 2018, or sorry, back in early January 2018, I'm sorry. And so the process that's shown here is that you collect the platelets and then you add amotosalen to it, and then you translate it to this new bag, and that's where you illuminate it with a UV illuminator. And then after that, you have to take out the amotosalen, all the amotosalen that's not reacted to any sort of other nucleic acid. And then once you take out the excess amotosalen, then you put it into these final bags, and these are the final bags that go into the patients. It's a logistically difficult process to add on, but we did it. So we collaborated with the hospital partners, both medical and administrative leadership, and we had an interrupted and robust supply of PRT products. And we implemented, like I mentioned earlier, PRT platelet production back in January 2018. We started in small numbers, and we've been slowly increasing the PRT production over the years since then. This is just a quick chart to emphasize, to sort of illustrate how we had increased PRT production over the years. The top row is the pediatric hospital. The bottom row is the adult hospital. Really, there's three colors, but the red color is the regular platelet products that are not PRT treated, And then the green and blue are the PRT treated products. So as you can see, when we started in January 2018, it was very minimal. And then over the years, we've gradually increasing in a proportion. So by February 2021, we were actually at about 50% of the platelets being produced were actually PRT platelets, which we're really proud of. So, over the years, we've gotten used to it, and our hospital partners have gotten accustomed to using PRT products. And so the next step in this sort of evolution is actually going from the 50%, the 40 or 50% that we're making, to 100%. And that's something that we are very proud of. We recently optimized all of our platelet collection procedures and volumes, and also our PRT platelet production processes here, so that we can make not just 50%, but 100% of our platelets being PRT. Like I told you, the process is not straightforward. Well, it's straightforward, but it's not easy. So to be able to do this efficiently was a logistical sort of tour de force here at the Blood Center. We implemented 100% PRT in May 2021, so just last month. And we're very proud to be one of the first blood center of this size to have 100% PRT production of our platelets. And the thing underlying this, to keep in perspective, is really to decrease almost to zero, if not zero, the risk of bacterial contamination in the platelets. That's really what we, have in our sights, and I'm glad we're able to sort of do that and ensure that blood safety is at the highest level for our very vulnerable patients who use these products. Now, I sort of kept harping on this a little bit, right? PRT is good, but operationally, like I mentioned, it's a little complicated. There's many more steps involved than just making it, a regular platelet product. There's an addition and removal of that small molecule, right? And it's like nothing is 100%. This is really good, but nothing is 100%, and it's known to be ineffective against certain viruses, particularly non-envelope viruses, right? So it works on most clinically relevant ones, but there are a few that it misses. And furthermore, right now it's only approved for platelets. So you still have the whole other big gorilla in the room with the red cells. The red cells cannot be PRT treated at this moment because that technology is still being worked out for red cell treatment. Okay? So the question is there a better way to sterilize blood products efficiently, right? So sterilize blood products and do it efficiently. And so this is where the final topic comes in, using a linear accelerator to sterilize. Now, a couple words about sterilization, right? So in general, a sterilizing dose of irradiation is considered to be about 25,000 gray, 25 kilogray, right? And to put that in perspective, currently we irradiate a proportion of our products, but for a different reason, not for sterilization, but for a different reason. And that method of sterilization is only 25 gray. And so if you want to go from 25 gray to 25,000 gray using the current irradiation practices, it'll take 1 week for you to sterilize it. That is just not acceptable. The product would definitely go out of temperature as you keep it in the irradiator. So there has to be a better way, right? And so that's where this linear accelerator comes in. The linear accelerator can generate up to 25,000 grays, with processing and everything in less than 5 minutes. So that is potentially something that we can sort of harness and sterilize our products with. I honestly don't know if it's going to work. It seems, but this is science. This is why you try, and we'll see what happens. So we're currently, I'm collaborating with folks from the Stanford Linear Accelerator Center, faculty members from radiation oncology, and of course the Stanford Blood Center. This is a very nascent project, and I'm going to try to see what effects sterilizing doses using the linear accelerator will have on platelets, RBCs, and plasma. We're going to test it for product viability, and stay tuned. I don't know if it's going to work. I honestly don't, because that's a massive dose of sterilizing radiation that you apply to an organic product. So, but if it does work, I think that'd be super, super exciting. And with that, sort of cliffhanger. I'd like to sort of open it up for questions at this point.
Thank you, Dr. Pham, for that informative presentation. We've got a few questions coming in. Zeke and Jane ask, can you elaborate on how amoxicillin works?
Amoxicillin? Yeah.
You pronounced it better than I did.
Yeah. I think I had a slide that went over that process. Maybe that question was prompted before that slide? But so amoxicillin is a small molecule, and it looks, if you draw out the picture, if you draw out the structure, it kind of looks like a nucleic acid. And the thing about one of its properties is that it can intercalate within the DNA molecule or RNA of sort of neighboring nucleic acid, and it will just kind of park itself there. And it's also highly reactive to UV irradiation. So as soon as once it parks itself next to any sort of nucleic acid that's in the product, you irradiate it, and the amoxicillin would cross-link itself with any neighboring sort of nucleic acid it can find. And once it cross-links it, it's a covalent link, so the nucleic acid cannot be replicated.
Thank you for that. Nancy asks, what, in your opinion, has been the most significant achievements in blood testing over the past decade?
Oh, the past decade. Oh, wow. So I think one of the best ones, so nucleic acid testing, I think, was a very big step. That was implemented in early 2000s, so it was a little after the last decade. But I think that was like a very big step, because now we're not looking at serology only, because like I mentioned, serology, the antibody test, there's that 2-week window where you could be infected and we can't detect anything. With the nucleic acid test, you can actually test for the bugs themselves, whether they're present in the blood at that moment.
Thank you for that. Bob asked, did the COVID-19 pandemic affect the way Stanford Blood Center tested donations?
That's a great question. So for all these tests that I listed, the short answer is no. We kept testing them the way that we tested them. Now, at the beginning of the COVID-19 pandemic, it was unknown whether COVID could be spread by blood, right? A lot of things were unknown, and everybody just assumed the worst, or most people assumed the worst, and most were right. But one of the things that we had done here was we tested all of our donations for the SARS-CoV-2 virus, the virus that causes COVID-19. And we actually did that for like 7 or 8 months. We tested 20,000 donations serially every day just to make sure we can't find that virus in the blood. And of like over the 20,000 plus donations that we tested, only one was positive, and it was a very low positive, and we discarded that, of course. But after that, we felt like, we felt that the data that we had was supportive of the fact that SARS-CoV-2 does not circulate in the blood that well, and if it does, it's probably very low levels. And also towards the end of that, other groups are also finding supporting evidence that SARS-CoV-2 or COVID-19 is not a blood safety issue.
Thank you for that. Harpreet asks, at this time, because PRT is not universal for all blood products, would you categorize the primary impact to be bacterial contamination?
The primary impact, the primary benefit of PRT right now, I do believe is bacterial contamination, because that, like if you look at that table of TTIs, that's sort of the highest risk at this moment. Yeah.
Daphne asks, do you test each donation individually or in batches?
Oh man, great questions, great questions. Get into the weeds. So certain tests, we test in pools of 6. So many of the nucleic acid tests that we do, we test in pools of 6. Many of the serology tests that we do, we test individually. And like if the nucleic acid test, we test in pools of 6. If the pool is positive, then obviously we retest the individuals in that pool individually to figure out which one is positive. But that's the answer.
A follow-up from Harpreet. If there is an emerging pathogen that may impact the blood supply, do you anticipate fast-track approval from the FDA to use universally? How fast could this be operationalized?
Oh, to use universal what?
I'll repeat that. If there is an emerging pathogen that may impact the blood supply, do you anticipate fast-track approval from the FDA to use universally?
To use universally?
And a follow-up saying, how fast could this be operationalized?
What's this that's being referred to? So maybe I'll just start a little bit and then maybe...
PRT,
Oh,
sorry.
PRT. Well, see, the thing about PRT, the reason why it's being used in platelets and plasma right now, but mainly platelets, is because it works, right? There's been a lot of studies going on with PRT being used to treat red cells. And right now, I don't know what the data are, but it's been worked on for a while. So I'm assuming they're still working on it, which makes me think that it's not up to par yet. But the thing about red cells is that hemoglobin makes a very dense shield of like a canopy almost, so that that step where you do UV illumination, it's hard for it to penetrate through all that hemoglobin. So that is my guess is maybe one of the obstacles of getting PRT treatment to work effectively in red cells right now. But I'm sure the folks at the Cirrus Corporation would know a lot more about that.
A follow-up from Zeke and Jane. In the PCR diagram you had, what does DNTP stand for?
Oh, eagle-eyed folks. So DNTP is just generic term for just you. So you need to give it the polymerase nucleotides to add to that growing chain. So the DNTPs is really just the A's, C's, G's, and T's that you would that would require to amplify the DNA.
Our final question from John. What do you think the next big news or hurdle will be in the world of testing blood?
Well, there's a lot of stuff going on with testing. Well, there's a lot of technologies right now, right? Like there's a lot of sequencing technologies out there that can test, that you can set up to test a lot of donors, a lot of people for a lot of bugs all at once. I imagine if, the graph I showed you, I kept talking about like testing, there's an upper limit because you can't test for every bug in the sun because every time you do a test it's for a specific bug. It's just not cost effective and resource intensive. But, I imagine that there will come a day when the technology will catch up and things will be cheaper so you can test everything all at once in one shot and not have to worry about individual tests. And maybe that would be the next sort of big thing, but that's purely speculative.
Anything else you would like the viewers to know?
Come donate. Yeah, we need that blood to keep on coming in. Thank you very much.